Okay, good morning. My name is David Carr. I'm manager for the Starry Stonewort Collaborative. We're based here at the Finger Lakes Institute at Hope Art William Smith Colleges in Geneva, New York. Also, we have, if you see there on the screen, we have the Finger Lakes Prism is hosted here. It's a partnership for regional invasive species management. We're one of 8 of those across New York State. Very briefly, the Starry Stonewort Collaborative has the general mission, I guess I'll call it, to bring together and increase the abilities and capacities of everyone from experts down through resource managers to citizen scientists to better understand and control Starry Stonewort across the Great Lakes Basin. So even though we're based here in New York State, this is a Great Lakes Basin-wide project. We've got over 23, 24 collaborators across the basin and 8 expert panelists that are helping us guide us through this process. They are critical to this program. We need their help, and we need everybody's help to get together and move this forward. And we really want to get all this reached down to the citizen scientists. We want as many people as we can ultimately in the field helping us identify correctly and reporting where Starry Stonewort is, and learn more about its life cycles and what makes it tick, because we need to know much more about it. Today, we are working with folks from Minnesota and Wisconsin. They have a really neat program that they do simultaneously in August every year. They train a number of people all across their state on identification of Starry Stonewort and other invasive species. So those folks are going to give us a quick overview today on those programs. So from Minnesota, they have what they call their Starry Trek program, which everybody I tell that thinks is the best name ever. They're all from the Minnesota Aquatic Invasive Species Research Center. You got Dan Larkin, and he's the kind of the lead in that area, I believe, and Megan Weber. And I got to slow. Oh, there were Patrick Mulcahy working with them as well. These folks are both Minnesota and Wisconsin are doing tremendous things, trying to understand and control and identify where Starry Stonewort is. And then from Wisconsin, their day is called the Snapshot Day, but again, these are sister programs, so they happen at the same time, and they'll tell you more about that. But Natalie Dutak or Dudek, she's from the River Alliance of Wisconsin, and Paul Skowinski, who is from the University of Wisconsin Extension Lakes program. He's at Stevens Point in Wisconsin. I've had the pleasure of working with Paul directly now, and there's some great stuff going on. I've talked to a number of these people on the phone. So with that, I turn it over to them, and they will enlighten you on this really neat training program they give every year in both states.
Fantastic, thank you. This is Natalie Dutak with River Alliance. I am the Aquatic Invasive Species and Watershed Groups Manager. And I'm really briefly going to go into the history of how Snapshot Day started and then how it evolved into this great partnership with Minnesota and Starry Trek. As an organization, River Alliance got involved with aquatic invasive species work about 10 years ago. We recognized that managing invasives on rivers is a pretty complex issue because there are multiple access sites along a river. It goes through multiple jurisdictions and states. And then of course that early detection is obviously one of the best ways to prevent the spread of invasive species. So we wanted to work on citizen science monitoring programs, and we developed one called Project Red, but around 2013, Laura McFarland, our then invasive species director, realized that it might have been a little too complex or required a little too much commitment from a volunteer, and that there also was another issue, which was detecting invasives at points where humans and the water interact. So on rivers, those were bridges, places where people might dump their aquarium species. And she wanted to find a way of creating a simpler program that could kind of get people out on the water at these locations, empower them and educate them to identify these invasive species, and then hopefully leverage that into future monitoring. So she piloted the first informal snapshot day in 2013 on the Kickapoo River. And she was really hoping to have this new way of getting citizens involved in stream monitoring. And in 2014, she partnered with Wisconsin Department of Natural Resources to create stream monitoring protocols and develop a way of selecting sites, priority sites, which we'll go into detail a little later, but are related to proximity to invasives, but also to those access areas where people might, again, you know, interact with the water and bring some invasive species with them. So we held our first official snapshot day. It was then known as AIS Bridge Snapshot Day, and that was in September of 2014. And since then, that name has slightly changed, and we've also obviously shifted the time frame to August. It was a better time to reach more volunteers. And then in 2017, just a year before I started, we began our partnership with UW Extension and Minnesota Extension to work on with Paul to make the lakes protocols, and then eventually to partner with Starry Trek. But I am not the best person to discuss that, so I will take a step back.
Hi everyone, this is Dan Larkin from University of Minnesota, and I will give an overview on this session. So, as Natalie mentioned, in 2017, Snapshot Day and Starry Trek joined forces. And this one-day event, the sister programs, as David mentioned, in Minnesota, we call it Starry Trek, and we're focused really on Starry Stonewort. In Wisconsin, it's AIS Snapshot Day, and it's broader, encompassing Starry Stonewort as well as other aquatic invasive species. And both events run concurrently on a single day in August. So, you know, early detection of invasive species is really critical. It provides the greatest potential to reduce impacts of invasive species, to prevent further spread, and it gives the broadest palette of options for control and the highest likelihood of success for control efforts. So that was really the underlying motivation for this effort. And there's high uncertainty in early invasions like Starry Stonewort throughout the Great Lakes Basin, and certainly in Wisconsin and Minnesota. So for example, what is the actual distribution? What is the potential for further spread? What are the potential ecological and human impacts of the invasion? There's uncertainty around all of these issues. And for that reason, there's a need for increased surveillance capacity. And so our goal was really to harness the power of professionals and interested members of the public to work together to get more trained eyes on the water for early detection. And Starry Stonewort, as we're all interested in today, is one example of a good place to apply this type of approach. It's a relatively new invader, and it's an increasing concern for AIS management. This is from a paper that Paul and I and others worked on together, and this is showing the history of Starry Stonewort invasion in North America from the early 1970s, when it was first found in Quebec, up until '78, when it was found in New York State. And you can see there was this kind of gradual buildup, and then in the mid-2000s, it started to be found more frequently. And part of that is, you know, greater awareness and greater search effort for this cryptic algal invader. And then in 2014 and 2015, it was found in Wisconsin and Minnesota, and you can see there's been this real increase in detection of Starry Stonewort populations. And here we're showing both a line for populations that have been confirmed, good vouchering and expert identification, as well as just total reports. And the difference between those is driven largely by Michigan, which has the most Starry Stonewort, and where there have been many reports that haven't necessarily driven the type of attention and expert confirmation that is associated with new Starry Stonewort finds in states like Wisconsin and Minnesota that have a lot less of it. So this is the current known distribution when we published the paper, with some purple squiggles thrown in. Since then, some updates, and you can see very much in this Great Lakes Basin footprint that David described as their area of focus. And so for our states, this is where we know it occurs in Minnesota and Wisconsin, shown with those colored dots, eastern Wisconsin, and then sort of pockets in central and northern Minnesota. But where else might it already be? This is, again, a fairly cryptic species. Not a lot of people spend much time trying to identify charophytes. So it could be in many more places already, as well as new infestations that are emerging. So that led us to develop this collaboration. This August will be our third time holding this event, this 2-state, 1-day search effort. And we have search locations that are prioritized based on the input of our host coordinators, who you'll hear more about. Prediction models, so Ranjan Muthukrishnan, who was a postdoc with me, developed these risk maps for Starry Stonewort invasion based on water chemistry, and then Wisconsin DNR has developed priority maps for their search efforts. And here, this map is just showing lakes that we predicted to be higher risk based on water chemistry. And now I will turn it over to colleagues in Wisconsin to talk about their site selection process.
Yep, I can take this one. In Wisconsin, we have protocols and spreadsheets that are sent out by the Wisconsin Department of Natural Resources, and they use criteria like unverified records where things are reported but not confirmed, and locations within 15 miles of where a prohibited species has been found. So these are species that are in very low numbers or not present at all in Wisconsin. If we see one of those pop up somewhere, then within this map, there's a 15-mile buffer that's drawn around these sites. And we try to pick sites within that buffer for Snapshot Day. And we also take into account known distribution of aquatic invasive species. So again, selecting sites that are nearby to where those AIS are known to be. And we also incorporate access information. So we're making sure to send people where there's adequate access for Snapshot Day activities.
Great, so this is Megan over on the Minnesota side again. I'm going to chat a little bit then about kind of how we organize the event and the things that we provide versus what we expect local coordinators to provide. And most of this is the same across the 2 states. Where there's differences, I'll try and call those out. But in general, we provide equipment. So we'll have vegetation monitoring rakes. We usually have free tote bags each year that we send out to each of the sites for volunteers to take home with them. We always have boot brushes in there so that volunteers who are out can be sure that they're cleaning their footwear and equipment before they're moving around or leaving that site or heading to a new one. Drying rags to clean off rakes or anything else that's had water contact, and an invasive species ID sheet. The ID sheet is usually a little bit different between the 2 states, again, because we have a slightly different focus on, narrowly on Starry Stonewort in Minnesota versus a broader suite of invasive species in Wisconsin. There you go. And then for the other thing that we provide is logistical support for those local coordinators. So we're kind of the statewide coordinators of the event. So on each side of the border, we each have our own statewide registration site. I think both of us use Eventbrite for that, but Minnesota has its own registration page and Wisconsin has its own. We host a training webinar. I put a link up there if you want to mark it down. You can see what that looks like, but We host a webinar each year. We've played both with doing them together as a Wisconsin-Minnesota joint webinar, and this year we recently tried a separate with Starry and Snapshot Day, each having their own webinars for their local coordinators. I think that link right now will move towards the one that we did just recently for Starry only. But they both have a similar layout on kind of the types of information that we try and provide on that webinar. And then we always record it so that anyone who can't make it can go back and view it later. And there's usually a matching handbook you can see at the end towards there where we give information about, tips for recruiting volunteers in the area, what the day of the event should look like. It has examples of the sign-in forms and data sheets and how they should be filled out. Contact info so that they know how to get a hold of the statewide coordinators if something comes up during that day. I think the handbook is really kind of built to be like the day of cheat sheet so that they can really have an easy, smooth run to the day. The other couple of things that we do is we help again with that coordination of the site selection. We have the standardized protocols, which for the lake monitoring protocols are the same in both Minnesota and Wisconsin. And then we help kind of statewide with event promotion. But then on the flip side, some of the things that we ask of our local coordinators is to, of course, be sure that they're reviewing the training materials that we're providing them so that it's kind of a cohesive event across each state, that they're going to host that site on the date that we set aside. So this year it's August 17th, and Minnesota and Wisconsin always tries to hold this on the same day. We usually have a rain date set aside as well, so ask them to hold, for example, the 17th this year is a Saturday, and we'll ask them to hold Sunday as well, so that if there's awful storms across the state or in parts of the state, those sites can delay by one day and host the event the next day. And then they train the volunteers, so we give them, the types of things that they need to be training them on, but they're ultimately responsible for making sure that they're going through that training on the day of. They have either direct data entry or data reporting in, and we'll talk about the two different methods coming up. Those are slightly different across each state based on what program the representative Department of Natural Resources uses. We really ask them to leverage their local networks where they know there's people of interest or where they know they have recurring volunteers, whatever, to make sure that they're also helping to bring local people into that event. They give us input on those locations, and then finally we ask them to do some printing, having refreshments like cleaning equipment, stuff like that at that local site for the volunteers. And then I think we're going to pop this over again for Paul and Natalie. They're going to discuss the protocols across lake and river monitoring sites.
All right, thanks, Megan. So the public access protocol we have for lakes is a little bit different than what we use for streams. And this, again, was developed in 2017 as Minnesota and Wisconsin started talking together about having this joint event. So when we send volunteers out to a lake access, we ask them to work in pairs or small teams, mostly for safety reasons. And they follow the protocol that I'll go through here in a second at each site that they go to. So we do often send them to multiple sites per team, and we ask them to decontaminate their gear between water bodies as well. We do tell them if they have a list of 2 or 3 or 4 sites that they're going to, that we do ask them, or we instruct them to go to the least contaminated site first, so that they're not going to an awfully contaminated, infested location first, and then potentially moving those things to new water bodies. So they do start with the cleanest sites first and decontaminate their gear between locations. So at the first location, they arrive and they identify the boundaries of the public access point. So this is going to vary quite a bit depending on the site. It could be very wide, it could be very narrow, but they look for the access points, boundaries on each side, and the surveying goes from one end to the other, and they plan out 5 locations between the access boundaries that are roughly equally spaced. And if there is a dock or a pier of any kind, we ask them to make that one of their sampling locations. If there's multiple docks, then we want all of those docks or piers to be sampling locations, because we know that boats are very commonly the main vector that move these invasive species around. So sampling off the piers is going to get us to the most likely places where these invasives are going to establish first. When they're on a dock, we ask them to sample off either side, but focus on those areas where the boats are probably moving through. So if they're clearly launching on one side, then we definitely want to sample over there to catch things that may fall off of a boat as soon as it's launched. These photos here show you air photos of just what a couple of different lake access points look like. So I believe there's some points that'll show up. Yeah, so the red marks there show you the public access boundaries. So that's what the site or the volunteer would first identify. And then they would plot out those 5 roughly equidistant points across that area. And you can see in the photos there that the docks are included as sampling points in every situation. So when they when they start sampling these 5 points at each location, they first scan the area for 30 seconds. We've noticed often somebody walks up to the lake and you focus so much on throwing a rake out to grab plants and things that you don't even notice that there's zebra mussels under your shoes, or there's crayfish, rusty crayfish or something crawling around right in front of you. So the first thing they do is just scan for 30 seconds, look all around them, look down, look around, see if there's anything blooming on the shoreline that is of concern, any large plant beds out in the water that they might want to target with their rake sample, or any shells or things that are washing up on the shore that are of concern. They also are instructed to look for plant fragments and things on the shore and use polarized sunglasses if they have them, since they are so effective at seeing into the water and spotting these things. And then at each location, they toss a sampling rake into the water, and we provide these rakes to them. It's a double-headed welded rake with the handle cut off and 35 feet of rope on a spool. I can't remember if Minnesota has the same length, 35 feet, but that's what we provide here. So it's enough room for them to get well out into the water and adequately sample what's there at that site. And we do tell them to aim for anything that you feel is suspicious. So during that scan time, if they found a bed of plants that they feel might be Eurasian milfoil or some other invasive that they're concerned with, then they would aim for it with the rake. And when they bring it back in, they pull material off the rake and use the ID resources that we provide to them to help identify any suspicious organisms. We do instruct them to collect anything that's suspicious, so we don't expect them to be able to identify everything on site, whether it's an invasive or a native species. Either way, we hope that they will recognize some things, but we don't put that obligation on them. So if there's anything that they think may be suspicious, they collect it and they bring it back for the local site coordinator to verify. So they put 1 species per bag, and they fill out a label that we provide to them that goes right inside the bag, filled out with pencil. So it's easy to figure out where those things came from. And then they turn those suspicious species back in at the rendezvous site to their local coordinator, and the local coordinator then takes the time to identify all these specimens that came back in, and they take care of verification and reporting and things later on. So we do ask them to complete their data sheet at each access point, and then once they're done with all their locations, then they turn all those sheets in to the local coordinator when they get back. And of course, they need to clean all their equipment and shoes after each site. So they're removing any fragments, they're rinsing stuff, and they're using a boot brush, which we provide to them. We have a brush that has a stiff bristles on one side and a pick on the other side, so they can adequately clean their equipment.
All right. This is Natalie. I'm going to quickly run through the stream or the river monitoring protocols. We have 4 different protocols, either dry for bridges and shorelines or wet for bridges and shorelines. And that can kind of address the different desires or needs of your volunteers, but it also allows site leaders to control when people are entering the water. For example, we had a site with New Zealand mud snails last year, and we used only dry protocols to keep people out of that infested water. So for our protocols, it's very simple. It's 2 minutes per step per transect, and at each step, your volunteer is going to look for emergent or riparian vegetation in the water and along the shoreline. Then they're going to spend another 2 minutes raking, and they're again looking for submerged plants and invertebrates. And then when possible, they will also spend 2 minutes scooping and going through substrate and looking for interesting critters in the water. And then they will collect those specimens, bag them up, and clean their equipment before moving to another site. And as Paul mentioned before, we also recommend that site leaders send their volunteers to sites of least contamination first. So everything is planned out to prevent that from happening. And yeah, and those are already mentioned the 2 different methods and the 2 site types, so I can go into a little detail on those. So for the dry bridge protocol, which is probably the most detailed, you have 4 transects. There's 2 on either shoreline and on both sides of a bridge. You would have your volunteers start standing within the right-of-way at the first transect, so transect A in this little drawing, at a location where most of the shoreline could be seen, and then they can scan the banks for 2 minutes. After that, they can approach the shoreline and using either a hard-handled rake or one of the rope rakes that were shown in Paul's slides, they can drag the rake through the water for about 2 minutes and collect specimens that way. And the specimens should be inspected after each drag of the rake. And then if it's possible, they can also use that handmade scoop to collect substrate and sift through. So once that's done, they will proceed to transects B, C, and D and repeat the 2 minute protocol for each of those sites. For the wet protocol, as the name implies, this time they are going to pretty much do the same thing, but they're going to wade into the river and collect samples at 4 transects spaced about 20 paces apart. And then once again, they're going to look for 2 minutes, rake for 2 minutes, and then scoop at several locations within the transect. So at each transect, they're going to be looking, raking, and scooping at least 3 times. And then they'll move, after each transect, they're going to move 20 paces downstream and repeat the process. For the shoreline wet protocols, they're going to, it's very similar. They're going to begin at the end of the reach at point A and then move upstream to point B. As with the bridge wet protocol, they'll have 4 transects spaced 20 paces apart, and they'll follow the same process as the wet bridge protocol. They will walk in or wade along the shoreline back to point A, being observant as they go, once again, just looking and scanning the shoreline on their way back. Shoreline dry is even simpler, and for this process, volunteers start downstream at the end of the reach at point A. They do their 2 minute look, rake, and scoop for specimens and substrate where possible. And then once again, they walk to the top of the reach, point B, while being observant as they go, and this time staying out of the water. And as Paul mentioned, everyone fills out a data sheet and cleans their equipment. That's it.
Okay, this is Megan again. So I'm going to bounce back to kind of what the rest of the day looks like on the event day. So this is the same across both states, kind of a general outline to how the day goes. From 8:30 to 9 is kind of usually when people are coming in. That's when they're signing the registration forms. We have liability forms on both sides for volunteers. They get their site assignments, get equipment and such. Then there's about 45 minutes that's set aside for training. So the local coordinator can use kind of whatever they have available. We'll provide what we can, and that varies across the state what we're able to provide. But it may be live specimens, there's scans, there might be images, there's again that ID guide, and we give kind of that step-by-step guide again on what all should be covered during that training period. And we always ask that they take group pictures too, so that we have those available to use in presentations like this or when we're trying to advertise the event elsewhere. So we always hope that we get in group pictures from each of the sites across the state. Then they all get sent out then to go to their routes or various monitoring sites and are back by noon generally is what we ask for, definitely no later than 1, but try and have people back by noon. So it's kind of a nice short-term commitment and they can still go and do other things for the rest of their Saturday. And then from 12 to 1 is when the local coordinator is doing their end of the work. So submitting data, working on the vouchers, having refreshments for volunteers as they come in, that kind of final bit of cleanup in that chunk of time. Some of the other details for the event days, I mentioned the liability waivers, so everyone has to sign those, and they're the same waiver across the state, different in Minnesota than Wisconsin, of course, because it's releasing each of the various host organizations, but it's the same waiver across all sites within a state. We only send people out to the public accesses, so we're avoiding issues of trespassing on private property if there aren't permissions or any kind of additional liability that might be associated with that. And then each of the volunteers, we're not expecting them to be pros at identifying, but we're hoping that they'll at least be familiar enough with the invasive species that they might encounter to say, like, oh, I think this is something that I should bring back for someone to take another look at. So that's when they follow that kind of bag and tag method that Paul discussed, of putting the species into the bag with the label and bringing it back for the coordinators to process in the end. So anything that's somewhat suspicious to a volunteer will come back to the local coordinators, and the coordinators do that next round of processing. Let's see, and then so for things that aren't needed, this varies by state, depending on what those protocols are. So I think this has mostly Wisconsin up here. So they're bagging invasive plants in bags that are labeled, and DNR has approval for landfill. Same Minnesota, we provide garbage bags, and they're allowed to landfill those. And then humane disposal for animals if they do come in. Let's see, and then I think Natalie is going to chat quick about what the Wisconsin data sheet and protocols look like, because this is another spot where it's a little bit different across each state. So I'm going to turn over to Natalie for this slide.
Sure, thank you. In Wisconsin, as we're looking for quite a few species, we ask, again, volunteers to look for anything, basically anything that they find, whether it's invasive or not, because again, they're not experts, and this is a great opportunity for them to learn. When they bring their data sheets back to the training site, this is where the site leader or the site coordinator helps them identify the species. And it's also where decisions are made on whether they're sending a verified voucher form in. And that's for specimens that are clearly prohibited species, if the species was not previously documented in a water body, or if the site leader actually is unsure of what the specimen is. And this year things are a little different because the Wisconsin DNR's regional AIS coordinators are actually working with the site leaders to help them verify either on the day of the event or within the week after. And then they can enter all of their data into SWIMS.
So on the Minnesota side, we do have data sheets similar to Natalie's for the volunteers to fill out that come back to the local coordinator. And we get those back as the U does as kind of central coordinators. And we use that just as information about what kinds of things they might be getting that they're confusing with invasive species. And it kind of helps us get an idea of, how to best phrase or target our educational items. But what the local coordinator is ultimately entering anything that they think is likely an invasive species or feel unsure about is EDDMapS, or the Great Lakes Early Detection Network, is the app associated with it. And this is what Minnesota has shifted to now. Minnesota DNR uses this as their database of invasive species records, both for aquatics and terrestrials. So this is an example from the app. You can see the type of information that gets entered into that. So most of this information the volunteer has on that data sheet for the local coordinator, and then once the local coordinator feels as though that's something that should be reported, they'll submit this information in using the app, or they can do it from a website from their office later if they prefer. One of the big things with it is images. So they take really good quality images, and these then get delivered directly to the DNR's AIS specialist for that region. And based on the images that they get, they will then make decisions on what they'll go follow up with and what the best follow-up method is. So if they need to go and do an in-person survey, the coordinator keeps as well the specimens that they sent in, so they can request that those specimens either get delivered to them or they'll go pick them up to do an in-person analysis, kind of whatever makes sense for the images that they get. But they'll get them on Monday when they get back in the office, and then they'll start processing through all the EDDMapS records that come in from the weekend.
We're talking a little bit about some of the successes and the results of our event. In 2018, we had 173 participants for our Snapshot Day, and 153 lake and river sites monitored. And if we break that out, it was 48 river and stream sites, 50 lakes, and 3 wetlands. And we worked with 20 partners. And the previous year, we had only had 125 participants and 139 sites monitored. So this was great. This is a great improvement. And this year, we also have closer to 25 partners. So we're hoping that number goes up this year and that we can verify more observed populations. I believe there's a map on the next page.
Oh, these ones might be out of order. There's a map, I think, next.
Yeah, there it is. And that's just those distribution of our Snapshot Day sites last year. Orange is the training site, and the blue dots are all of the monitored sites.
So this is Dan again from Minnesota, and this is showing our search effort so far for Starry Trek. On the map, the different colored symbols are from different years for the green and the blue, and then the red symbols are significant finds from the event. So in both 2017 and 2018, we've had about 20 local training sites or rendezvous sites with host coordinators. We had 200 volunteers in 2017, and then that number grew in 2018. And in both years, we've searched around 180 different water bodies, and somewhat more than that of public accesses. So some of the water bodies having multiple public accesses that we sent people to. And both years, there were new discoveries of Starry Stonewort done by our volunteers. And in 2017, for example, a population of Starry Stonewort was found on Grand Lake in Stearns County. And we view this as a real example of the power of this type of approach. The photo with the penny is what was actually found by the volunteers. It was a small fragment, but it was enough for them, based on the training they had gotten that morning, to think it was something suspicious that was worth taking a closer look at. It was confirmed as Starry Stonewort, and it was found at a time when it was quite a small population, restricted to one public access. So it was a real needle in a haystack type of lucky find that, you know, that population, who knows when it would have been found without this event. And what this early detection enabled was a partnership between Minnesota DNR and the Grand Lake Association, where DNR staff dove and carefully hand-pulled the population. And that's something they've continued to do. And so there's still Starry Stonewort there. It has not been eradicated from that location, but it has been kept in check. It has been contained, and it has not been found beyond the access area. And hopefully with continued sort of ongoing weeding, this population can be at least contained and potentially eliminated.
And similarly, we have a couple of nice success stories from Wisconsin as well. This is a particular site where water hyacinth was found. And this is an interesting situation in that it wasn't during Snapshot Day, but it was several weeks afterward when a participant of Snapshot Day, the first one, remembered the training that she had when she ran across a strange looking aquatic plant when she was paddling around on the lake near her home. And she was pretty sure it was water hyacinth, and she collected a sample and reported it, got a sample to professionals that were able to verify that species. And then shortly thereafter, we assembled a team of DNR staff and county staff to go out and help her to remove this population. And so you can see that pile there. That's actually last fall. So this population has shown up 4 years in a row in the same area of this lake. This is in Lake Winneconne, which is attached to Lake Winnebago, Wisconsin's largest lake. And what flows out of Lake Winnebago ultimately ends up in flows out of Lake Winneconne ultimately ends up in Lake Winnebago. So it is a big concern for us that this species is there and it keeps showing up. So we don't entirely understand whether this thing is actually returning on its own each year or if it's being consecutively reintroduced by somebody each fall. But the volunteer continues to participate in Snapshot Day. She continues to monitor that area of Lake Winneconne and report things to us immediately. So it's been a great success working with her and other participants that really get trained during Snapshot Day and utilize their training for the whole year, and they don't just kind of hang up their hat at the end of Snapshot Day.
So here is our contact information. If you'd like to follow up with any of us. And we also have some time to take questions as well. And Paul, I wonder if I should turn the presentation back over to you, or if you're able to see. I guess what I'm wondering is how we want to handle the Q&A.
If you want to send it back to me, that's fine. I think we have a fairly small number of people on the call, so it's probably okay if they just would like to unmute and ask the question. There's also a chat function at the bottom of your screen. You can click on the chat and you can send that to all participants, and then we'll all be able to see that and respond to written comments as well.
Okay, that sounds good. And if you could watch the chat feature with the screen share, I can't see those.
Sure. Yep. I'm watching it right now.
And Paul and Dan, this is Dave Carr again. If I could just say a couple words about the slide you just put up. Finger Lakes Institute's been really excited to be listening and talking and learning with all you folks about this. And as you know, we would like to build on this, use your programs as a model. And we want to use our program to expand that into other areas of the Great Lakes Basin. And that's a tall order. So as one of my coworkers says, you know, start small but think big. So we would love to get some folks involved, doing some sampling even this summer. We'll do some, if they're local to us, we'll provide some training. If not local to us, we will help find coordinators, site leaders, and we will, as you folks have done, we will supply training materials as needed, identification sheets. We're prepared to provide field sampling kits, you know, with rakes and a bag or backpack and identifying sheets or whatever is needed in the field. And so we would love to get some folks involved. You know, another thing the coworker said here, we're looking for small and mighty groups. If we get some sampling done this summer with a little training, just small groups, it's a start. I know the Minnesota and Wisconsin and Michigan have a tremendous amount of people out there looking at this stuff. But there's a lot of areas, other areas of Great Lakes Basin that are not looking so intently at Starry Stonewort, and we need to. And almost embarrassingly, because I run the program, our state, New York State's kind of behind compared to you guys. So again, if we can get something going this summer, just a small way, we will, and then we'll build this program to a larger audience. And then, of course, a program like what you just heard about, that's our target. You know, if, as I was talking to Paul recently, if we could have several other states, you know, the Great Lakes Basin do the same thing, everybody on one big day in August do a huge training thing, we'd be thrilled. That'd be a really neat thing to do. And that's our target. So please, as it shows here, that's our brand new, it's not even a full website yet. It's only 3 pages. But it's up, it's under construction, and it's going to be much, much more than what's there now. But there's three little pages there. One's a volunteer page to fill out a form if you're interested in getting involved in something like this. Give us some rough background information on your group or yourself, and we can contact you about it. We have a typical contacts page. You just fill in your information, and it'll be included in ongoing emails and news flyers, if you will, digital e-flyers. And then also there's a link there that'll take you to our also brand new Facebook page where we want to start building events and people using Facebook as a communication tool to talk back and forth about what they're doing, what can be done, and so on. So please take the time to contact me. Go to this website, contact me through that, whatever it takes. And again, we're going to start small, but we want to grow big, and we want to mirror what folks here in Wisconsin and Minnesota are doing. This presentation will be made available as well. Ultimately be on our website when the website gets to that point, but I can make it available to whoever other ways. And as questions are coming in, but I just want to thank everybody for sitting in here, and particularly thank all you folks in Minnesota and Wisconsin for all your good work and putting this presentation together. So I'll be quiet now. We start looking at questions.
All right, thanks, David. This is Paul again. We do have two questions that came in. I'll take the first one a little bit. Dan, I'll probably defer the second one over to you for your thoughts. The first one was, how does flow rate affect likelihood of starry stonewort infestation? And as far as I know, there is no research that looks at that. But as far as starry occurring in areas where there is substantial flow, there are a few channels in New York, especially where starry stonewort is established and doing just fine. So it can take a little bit of flow. In general, that whole family, the Cariaceae family that starry stonewort belongs to, and all the various native species that we have in that family, they do tend to like calm water. So flowing water, you find less of them as the flow rate increases, but starry stonewort certainly can occur in areas that do have some flow to them. And Dan, the second question was, oh no, we found starry. Now what do we do to control it, and how often?
Yeah, that's a good question. I think the first question is, how much did you find? So, the size of the infestation is really critical for determining what are viable options. So for example, here in Minnesota, the control efforts range from the Grand Lakes situation we described, where hand pulling can be a highly effective and selective method, to Lake Coronis, which was the first population that was found in Minnesota in 2015. And by the time it was discovered, it already covered about 200 acres of the lake. And so, at that point, clearly manual removal is not feasible. The most common control approach is copper-based algicide. And then there are also some non-copper herbicides that are used as well. Chemical control methods can certainly be effective for knocking it back, but it is it's really nuisance control. It's because of its biology, its production of bulbils that can sit in the sediment. Those are sort of like little tubers. They're asexual reproductive structures that can give rise to new plants. It's very difficult to eliminate starry stonewort using algicide, but it can certainly reduce the nuisance by killing back the biomass. But really, there's relatively little research that's been done on starry stonewort control. So in comparison to other species, say Eurasian water milfoil, for example, or curly-leaf pondweed or hydrilla, where there's been decades of applied research on control methods, we're really at a beginning stage of understanding how to control starry stonewort effectively. And so people are trying lots of different approaches, different algicides and herbicides, different frequencies of treatment. And so hopefully we'll be learning a lot in the coming years as these treatment efforts are monitored.
Yeah, and I'll just sort of echo that. In Wisconsin, we have several populations. We're close to a dozen species, a dozen locations or so of starry in Wisconsin, and we're looking at different treatment options for pretty much every site. So we do have one site that is not being treated. It's just a control at this point for the study, and most other sites are being managed in different ways so that we can evaluate different treatment options and hopefully come up with some sort of an answer or management recommendations in the future. I don't see any other questions in the chat at this time, so maybe we'll just wait another second here and see if any more questions come in.
Okay, this is Dave Carr. I think we're good. Is there any more questions? Now's the time, but you can always contact all of us in other ways via email and so on. And again, I want to thank everybody for participating, and particularly the Minnesota and Wisconsin folks. This is an amazing program you put together. And again, we're trying to model it. So we'll keep at it and see what we can pull off in other areas.
Thanks everyone for joining us, and thank you, Dave, for organizing this event and inviting us to present.
Thank you.
And I will now, so the recording feature.
You can end the meeting, and David's recording will automatically stop when you end the meeting.
Okay, because I can.
I'll
Okay.
stop sharing.
Or you can stop recording now, David, if you want.
Whatever, I'll do that.